So today we are discussing about the histology and the animal tissue culture. Class. Thank you. Thank you. Here you can see the anatomy. Here you can see the anatomy of the tissues. When coming to the study of the tissue, there are four types of tissues here: connective tissue, epithelial tissue, muscle tissue, and nervous tissue. As we know, tissue means a group of cells. Today we are discussing about the histology and microscopic etiology. It is the introductory part to our next topic after the introduction to the biotechnology. Here, as just we discussed, there are four types of tissues. And that is connective tissue, epithelial tissue, muscle tissue, and nervous tissue. And the study of tissues, the whole body contains 100 different types of cells. Histology, microscope, anatomy, the study of tissues and how they form the organ. Histology means especially the microscope, the study of tissues and how they form organs. Organs are the structures with discrete boundaries. A group of tissues is composed of to produce the organ. Organ means a group of tissue. It is composed of two or more tissue types. Differences among the four tissue classes, types and functions of cell. For example, epithelial and 3D, nervous and muscular. What are the characteristics of matrix? Extracellular matrix, rubbery and stony and gelatinous, and the relative amount of the space occupied by cells versus matrix, PG versus muscle and epithelium. What are embryonic tissues? Here, the embryo begins as a single cell, divides into many cells that form layers. Three primary germ layers: the ectoderm means outer layer, which gives rise to epidermis and nervous system. Endoderm, inner layer, mucous membrane, and gastrointestinal tract, and respiratory lining, digestive glands. Mesoderm, that is middle layer, forms mesenchyma, gelatinous tissue, and it gives rise to muscle, bone, and blood. What are epithelial tissue? Here, epithelial tissue, one or more layers of closely adhering cells at the top forms a flat sheet with the upper surface exposed to the environment or an internal body, that is cavity. Bottom on basement membrane, basal surface of the cells acts as epithelium to underline the connective tissue. Here you the structure. The structure of nerve cell. Here you can see the presence of basal surface and apical surface, lateral surfaces, and both the apical surface and lateral surface and the basal surface combinedly called as epithelium. And basement membrane contains basal lamina and a reticular lamina. And here you can see the presence of connective tissue. And what is epithelial tissue? arrangement and cell shape. The first, the simple epithelium, which contains one layer of cells, are named by the shape of cells, and stratified epithelium contains more than one layer, named by shape of the apical cells. Here you can see the shape of cells with the simple epithelium, and in this stratified epithelium, here you can see the base of the membrane. Here, the arrangement of the layers you can see a single layer and multi-layered. And when coming to the cell shape, here the cell shape is squamous and cuboidal and columnar. After seeing this picture, you can imagine a simply, you can differentiate simply after seeing these pictures, squamous, cuboidal, and columnar, which is connected with the basement. And what is pseudo-stratified epithelium? When coming to the structure, single row of the cells, not all of which reach the free surface nuclei at the differential level. And when coming to the functions of the pseudo-stratified epithelium is which secretes the propel the mucus. And the location, most of the upper respiratory tract system from nasal cavity to bronchi, a part of male urethra. When coming to the keratinized stratified squamous cells, here you can see the dead squamous cell, and the living squamous cells looks like this, and this is the connective tissue. Layers of epithelium covered with the compact and squamous cells packed with protein, that is keratin, and retards water loss and prevents entrance of the organisms from epidermal layer of the skin. And non-keratinized stratified squamous, here the epithelial layer you can see a multi-celled epithelial layer, a multi-layered epithelial layer you can see that lacks the surface layer of dead cells, forming moist stem cell, stratified cuboidal epithelial cells you can see in the structure of the structure two or more layers of cells. Those are surface cells, square or round shape. Functions are uh, secretion and production, and sweat glands, mammary glands, salivary glands, ovarian follicle, and seminiferous tubules. Transitional epithelium. When coming to the structure of transitional epithelium, it is a multi-layered epithelium with rounded, not flattened surface cells. And what are the functions? It allows the stretches and distension. So, where we can see that transitional epithelium cells means in the urinary tract, a part of kidney, ureter, and a urinary bladder, a part of urethra. Here you can see the relaxed transitional epithelium. Here you can see the multi-layered transitional epithelium with apical surface, a basement membrane with connective tissue. What is connective tissue? When coming to the connective tissue, in the last slides we discussed like a CTCT, connective tissue. What is connective tissue? Here, most abundant and variable tissue type. The functions of the connective tissue is a name itself indicating that which connects to the body. Uh, where it connects means The binding organs, for example, a tendon connects to the muscle to the bone, and the support, protection, and movement. Example, bones, and the storage of fats, bones, and transport. For example, a blood. A ground substance of connective tissue, gelatinous or rubbery material found in between two cells. What are these? Consisting of three classes of larger molecules. Those are glucosaminoglykans, AGS, and polysaccharides that attract sodium, which hold the water, and proteoglycan is a bottle brush-shaped molecule and forms thick gel that slows the spread of pathogens. A cell-adhesive glycoproteins allow themselves bind matrix elements. How the connective tissue, fibers of connective tissue, collagen fibers are called white fibers, most abundant protein of the body, thick, tough, and resist stretch and flexible. For example, tendons, ligaments, and that. Uh, Apart the collagen fibers, we have yellow fibers. Those were elastic fibers. These elastic fibers recoil like a rubber band, which shows the elasticity that can able to stretch. For example, you can see that yellow fibers in the skin, and it is also useful for lung expansion and contraction. So you can see that elastic fibers in lungs and heart arteries. This has the ability to recoil. And the other one is reticular fibers. This is the thin collagen fibers coated with glycoprotein. For example, form framework for spleen and lymph. Here you can see the structure of a hand where you can see the collagen and tendons, collagen. Here you can see the collagen. And what is the use of these collagen fibers means? These called white fibers, most abundant protein of the body, which is in a thick, tough, and that can resist the stretch and flexible. Though it is flexible, but that can resist the stretch. Where we can see that means tendons, ligaments, and that. Here you can. And what are the cells we can see in the connective tissue? Fibroblasts. A fibroblast produce fibers and ground substance, WBC, white blood cells. Those can wander mostly in connective tissue in search of bacteria. The white blood cells in turn is classified into two cells. Those were macrophages and plasma cells. What is the duty of macrophages means? A large phagocytic cells arise from the monocytes, which functions as a phagocytosis. And plasma cells which arise from the lymphocytes, the antibody producing cells. Mast cells, these are the oval-shaped cells clustered along blood vessels, secrete heparin and histamine. These two, heparin and histamine, that can produce, that is useful for converting the fibrin into fibrinogen. So these mast cells is also comes under the cells of the connective tissue, which repairs the wound. When coming to the adipocytes or fat cells, the duty of adipocytes, the main function of adipocytes or fat cells are store triglyceride. When coming to the first connective tissue, fibrous connective tissue, these fibrous connective tissue have 5 types of fibrous connective tissue. that divided into two broad categories. One is a loose connective tissue and another one is dense connective tissue. What is loose connective tissue means? These tissue contains more gel-like ground substance between the cells. There are three types of cells you can see here. That is alveolar, reticular, and adipose. You can see alveolar in the lungs and adipose just now we discussed and reticular in heart. And density, dense connective tissue, fibers fill the spaces between the cells. Two types varying in fiber orientation. One is dense or regular and dense irregular. See, just now we discussed alveolar loose connective tissue. Here it is. Here it contains the ground substance. Looks like this. The loosely arrangement of collagenous tissue you can see here, and collagenous fibers and the elastic fibers and the fibroblasts. The loose arrangement of collagenous and elastic fibers scattered the cell sites abundant ground substance. The locations underlying all epithelial and surrounding nerves and blood vessels, esophagus and tract. When coming to the reticular tissue, this reticular tissue comes under the dense, uh, loose connective tissue that is forms a structural supportive stoma for lymphatic organs. Here you can see the presence of leukocytes and reticular fibers. So these reticular fibers you can see in the kidney, lymph nodes, a spleen, thymus, and bone marrow. When coming to the adipose tissue, that is fat tissue, a large, empty looking cells dominate with the thin margins, a nucleus pressed against the cell membrane, often very pale. What is the function of this adipose tissue? Energy storage, insulation, space filled as cushioning. Locations, subcutaneous fat, beneath skin, skin breast. Under the skin, in the breast, we can see the presence of adipose tissue and the heart surface and surrounding organs. Here you can see the blood vessels and the adipose tissue in between the cells and the lipid in the adipose tissue, adipocytes. See, here we have taken the example of breast. And when coming to the dense regular connective tissue, this connective tissue mainly densely packed and parallel fibers, compressed fibroblast nuclei, scanty open space, and blood vessels. Allocations are tendons and ligaments. A dense regular connective tissue, collagen fibers, a ground substance, and fibroblast nuclei. Here you can see the presence of a dense a regular connective tissue in the leg. A dense regular connective tissue, a densely packed collagen fibers are running in different directions. A scanty open space, few visible cells and blood vessels. What is the function of this dense irregular connective tissue is withstands stresses applied in many different directions. The locations are deeper portion of the skin, capsules, around the organs, liver, kidney, and sheaths around the cartilages and bone. When coming to the dense, irregular connective tissue, You can differentiate simply by seeing these two pictures. A densely regular connective tissue with collagen fibers, and the arrangement you can see here, ground substance and fibroblast nucleus. And here the irregular arrangement of a ground substance you can see, and also the bundle of collagen fibers also you can see in an irregular manner with fibroblast nuclei and gland duct. And other connective tissues, those are cartilage, bone, and blood. When coming to the cartilage, the supportive connective tissue with the rubbery matrix, chondroblasts produce matrix, surround themselves and become chondrocytes. No blood vessels it contains, so diffusion must bring in nutrient and remove the waste. There are three types of cartilage depending upon the fiber types. Those were hyaline, elastic, and fibro cartilage. Here you can see the perichondrium, chondrocytes matrix. Here you can see the skeleton of the fetal, where you can notice the perichondrium, chondrocytes, and matrix. The arrangement of perichondrium and elastic fibers and chondrocytes in the ear pinna, the external ear, a low ear lobe. Here you can see the presence of perichondrium, the arrangement of perichondrium, elastic fibers, and chondrocytes. Here you can see the presence of chondrocytes and collagen fibrils and lacuna in the vertebral disc. When coming to the bone, osseous tissue, a spongy bone looks like spongy in appearance, feels heads of long bones, delicate struts of the bone, always covered by the compact bone. The compact dense bone looks solid, no space visible to the naked eye. External surfaces of all bones. Here you can see the presence of lacuna and concentric lamella of osteon, a central canal, and osteon that is presented in the compact bone. When coming to the back blood, a variety of cells and cell fragments, some with nuclei and some without nuclei. RBC, WBC, and platelets are presented in the blood, found in heart and blood vessels. Platelets, neutrophils, lymphocytes, erythrocytes, monocytes you can see in this single picture when blood is smeared. You can see the simple difference. You can differentiate the platelets and neutrophils and lymphocytes and erythrocytes with monocytes. You can see the nuclei of a particular cells. And when coming to the muscular tissue, alternating contraction and relaxation of the cells, the chemical energy changed into the mechanical energy. When the bone is transverse plane, here you can see in the image, three types of muscle tissue you can see. Skeletal muscle attaches, the skeletal muscle which attaches to the bone, skin or fascia, and straightened with light and dark bands visible with scope. Voluntarily, the control of contraction and relaxation takes place. Here you can see the skeletal muscle structure And three type of muscle tissue. One is cardiac muscle. That is the cardiac muscle appears in a straighted position, involuntary control, and autorhythmic uh, because of built-in pacemaker. When a person is connected to a pacemaker, here uh, whichever the autorhythmic cardiac muscle function is there, that is controlled by the pacemaker. And other one is a smooth muscle. Here you can see the smooth muscle. This is smooth muscle attached to hair follicles in skin, involves of halo organs. Those are blood vessels and gastrointestinal tract, and non-striated in appearance, involuntary. Functions of muscle tissue. What are the main functions of the muscle tissue is producing body movements, stabilizing body positions, regulating organ volumes. Bands of smooth muscle are called as sphincters, and movement of substances within the body. This is helps for the movement of substances within the body. Those were blood, lymph, urine, air, food, and fluids, and sperm. And the other function of muscle tissue is producing heat. This happens like involuntary. These are involuntary contractions of skeletal muscle, that is shivering is takes place. When coming to the connective tissue components, here in the last picture also we have seen this structure. Here you can see the structure of myoblast and satellite cell. These are the myofibrils in the muscle fiber. So the muscle fiber or the myofiber is having the satellite cells, sarcolemma, myofibrils, here you can see, and sarcoplasm. So what are these? What is the sarcolemma? Here the sarcolemma is a muscle cell membrane. Here you can see the cell membrane of the muscle is called as sarcolemma, and muscle cells are long, cylindrical, and having the number of cells, the number of nuclei. Uh, so it is called as multinucleated, a long and cylindrical shaped cell, which contains the sarcolemma and also sarcoplasm. Here you can see the presence of sarcoplasm. A sarcoplasm filled with the tiny threads called myofibrils and myoglobin. Myofibrils are in red in color and oxygen binding proteins. So here you can see the presence of myofibrils. This is the myofibril. So as just we discussed, myofibrils are red in color, oxygen binding protein. Here also you can see the presence of myofibrils, which, and here you can see the sarcolemma. And transverse tubules. What are transverse tubules? This is also called as tubules. Those contains the terminal cisterns, and uh, these transverse tubules are imaginations of the sarcolemma. As just we discussed, what is sarcolemma? Sarcolemma is the muscle cell membrane. So in the muscle, imaginations presented in the muscle cell membrane is called as transverse tubules, T tubules, which is filled with extracellular fluid, which carry muscle action potentials down into the cell. Mitochondria lie in rows throughout the cell near the muscle proteins that use ATP during the contraction. Here you can see the structure of transverse tubules, which contain mitochondrial, sarcolemma, thick filament, thin filament, and myofibrils. Here you can see the presence of myofibrils and sarcoplasmic reticulum. When coming to the structure of myofibrils and myofilaments, as just we discussed, myofibers are filled with threads called muscle fibers are filled with threads called myofibrils separated by sarcoplasmic reticulum. Myofilaments thin and thick filaments are the contractile proteins of the muscle. Sarcoplasmic reticulum, uh, this sarcoplasmic reticulum, a system of tubular sacs similar to smooth endoplasmic reticulum in non-muscle cells, which stores calcium ions in a relaxed muscle, which release of calcium ions triggers muscle contraction. So the main use of sarcoplasmic reticulum is uh, relaxing the muscle and contracting the muscle. So this is the sarcoplasmic reticulum. This is the place where the calcium ions storage taken place, and this is the place where the release of calcium ions triggers the muscle contraction. So, a system of tubular sacs similar to smooth endoplasmic reticulum in non-muscle cells are called sarcoplasmic reticulum, which contain mitochondrial, sarcolemma, thick filament, and thin filament, and it is also contains myofibrils and sarcoplasmic reticulum. Filaments and sarcomere, thick and thin filaments overlap each other in a pattern that creates the bands, light I bands and dark A bands. They are arranged in compartments called sarcomeres, which is separated by the Z disc. In the overlap region, 6 thin filaments surround each thick filament. Here you can see the presence of Z disc and M line and Z disc, completely called as sarcomere. And rigor mortis is a state of muscular rigidity that begins 3 to 4 hours after the death and lasts about the 24 hours. After the day, the calcium ions leak out of the sarcomere and allow myosin heads to bind to actin. Since ATP synthesis has ceased, cross bridges cannot detach from the actin until proteolytic enzymes begin to digest the decomposition of the cell. Neuromuscular junction or synapses. Here, myoneural junction, that is a neuromuscular junction. The end of the axon nears the surface of a muscle fiber, and its motor end plate region remains separated by the synaptic cleft or gap. Here you can see the structure of NMJ, neuromuscular junction. Here, the synaptic end bubbles are swelling of axon terminals, and end bubbles contain synaptic vessels filled with acetylcholine. Motor end plate membrane contains 30 million ACH receptors, that is acetylcholine receptors. The events which occur after a nerve signal: arrival of nerve impulse at nerve terminal causes release of acetylcholine from synaptic vessels. Acetylcholine binds to receptors on muscle motor end plate, opening the gated ion channels so that sodium ion can rush into the muscle cells. Inside of the muscle cell becomes more positive, triggering a muscle action, a potential that travels overcome the cell and down the teeth. The release of calcium ions from SR is triggered and the muscle cell will shorten and generate force. The acetylcholine esters break down the acetylcholine attached to the receptors on the motor and plate, so the muscle action potential will cease and the muscle cell will relax. When coming to the anatomy of cardiac muscle, here you can see the anatomy of the cardiac muscle, the presence of mitochondria and sarcolemma and nucleus and cardiac muscle fibers. This is the gap junction between the cardiac muscle fibers and the intercalated discs where the gap junction is filled, and desmosomes, capillary tubes, and straighted, short, quadrangular shape branching fibers are the cardiac muscles, and a single centrally located nucleus here. Here you can see cells connected by the intercalated disc with gap junctions. Same arrangement of thick and thin filaments as skeletal. So when you see the muscle under the microscope, here the smooth muscle looks like which containing visceral, a single unit, a smooth muscle. Here you can see the presence of blood vessel also. This is the transverse section and this is LS, longitudinal section. Here you can see the uh, difference between the transverse section and longitudinal section. Uh, Here the smooth muscle looks like with a small involuntary muscle cell with the tapering at ends and a single oval and centrally located nucleus. Here you can see a lack of T tubules and have little uh, SR for calcium storage. When coming to the microscopic anatomy of smooth muscle, here you can see the myofilaments, thick and thin myofilaments. Here you can see not orderly arranged. So the lack sarcomeres. Here you can see the difference between the relaxed and contracted smooth muscle. And just to be discussed about the smooth muscle, it contains the sarcolemma, intermediate filament, and dense body with the nucleus. When Smooth muscle is relaxed. Here you can see the structure. When it is contracted, here you can see the structure. And it contains the thin and thick myofilaments, not orderly arranged. It lacks the due to it lacks the sarcomere. And sliding of thin and thick filaments generates transfer and transfer to the intermediate filaments and dense bodies attached to the sarcolemma. A muscle fibers contracts and twists into a helix as it shortens, relaxed by un. When coming to the nervous tissue, nervous system, we have central nervous system and peripheral nervous system. When coming to the central nervous system, we have brain and spinal cord. When coming to the peripheral nervous system, the nerves and their associated ganglia. A microstructure of neuron. This is the microstructure of neuron. Here we can see the presence of axon and dendron and dendrites. A presence of peripheral nervous system. Here you can see. And we have head, the cell body, which contains dendrites, and the middle part we have the axon. A neuron, which possess the cell body and process of neurites. The cell body is divided into cell membrane, cytoplasm, and nucleus, whereas the process of neurite is divided into dendrites and axon. Uh, the cell body is also known as perikaryon. The cell body's soma is the part of the neuron that contains nucleus surrounded by the cytoplasm are also called as perikaryon. It is the trophic center of the neuron. The protein and enzyme synthesized in this area. And cell body of the perikaryon in the cell body of the perikaryon where the position and the shape and the size of the perikaryon is only in gray matter in the central nervous system, which also contains dendrites and axons starting from or ending on the cell bodies. Those are ganglia in PN. The shape of this one is they can be pyramidal, spherical, ovoid, or pear shape. The size of the perikaryon is measuring 5 to 150 micrometer in diameter. You can see this perikaryon under the microscope also. The cell membrane, the structure is as same as the normal cell. It functions in the getting the stimuli and integration and conductive the nerve impulse. The nucleus is large and pale in color with HE, hematoxylin stain. Prominent nuclei are very clear. Here you can see the presence of nasal bodies, nucleus and nucleolus and neuroglia and nuclei. This is the structure. And when coming to the cell body, when coming to the cytoplasm has some distinctive characteristics that not clearly visible in other cells. The cytoplasm is basophilic and full of neurofibrils. Here you can see the difference between the neurofibril and distal body. With the silver nitrate staining and hematoxylin stains. Nasal bodies, what are nasal bodies? The cytoplasm shows the presence of granular material that stains intensely with basic dyes. This material is the nissel substance, are also called as nissel body or nissel granule. And the neurofibrils you can see are thin black fibers absorbing LM with silver nitrate cells, slides, which is composed of microtubules and filament in electronic microscope. This is the structure. Rough endoplasmic reticulum and synapses, ribosomes, a pigment, mitochondria, and micro. Neurites are process. What are neurites? The process arising from the cell body of the neuron are called a neurites. Most neurons give off a number of short branching processes are called dendrites and one longer process called an axon. Here we also have the collaterals. Dendrites are characterized by the fact that they terminate near the cell body. They are irregular in thickness and cells granules extended into them. They bear numerous small spines which are of variable shape. When coming to the axon, every neuron has only one long in process called axon, which arise from a special region or axon hillock, which is devoid of cells bodies. It carries the impulse received by the neuron to distant region. An axon may have not much branches than that of dendrites. If branches that arise near the cell body and lie at right angles to the axon called collaterals, and at its termination, the axons break up into a number of fine branches called phylodendria, which may end in small swellings, terminal boutons. The axon is identified according to the axon hillock with LM, the part of the axon lateral axon just beyond the axon hillock are called as the initial segment. Neurites are the process where we can see the dendrites. Here we can see the axons. Here the what we call the presence of many, and the axon is only one, and dendrites are short in structure and axon is very long, and the dendrites are in irregular in shape in thickness. It is having the uniform in diameter and structure and diameter. Dendrites contains nissel granules. In axons, there is no nissel substances. Dendrites composed of spines. It is from axon hillock. This originated from axon hillock. Impulse towards the stoma away from the cell body. Dendrites always impulse towards the cell body, that is soma. Axons away from the cell body. Identification of dendrites from the axon. Dendrites have microtubules associated with protein MAP2 in immunocytochemically not present in axons, which is distinguished from the axon. The classification of neuron according to the number of processes. A neuron is a unipolar or pseudo unipolar polar, and bipolar and multipolar neuron. That is based on the number of processes. Here you can see the structure of unipolar, and here you can see the bipolar presence of two dendrites in the each end. Here the neuron contains only one dendrite, and multipolar neuron. Here multipolar neuron that is there in Purkinje cell, and also in pyramidal cell, and smooth muscle also. Here you can see the unipolar neuron in the spine ganglia, and bipolar neuron in retina of the eye, axon hillock, a multipolar neuron, and here I want to explain. Here I want to explain the general histology and levels of structural organization. Just now, this is just like a summary. The atoms combine to form the molecules. The cellular means the cells made up of molecules, and tissue consists of similar type of cells. The organ which is made up of tissues, organ system which is made up of different organs that work closely together. When coming to the organismal, which is made up of number of or organ system is called as organism. Here you can see the level of structural organization. As just we discussed, a chemical level of atom combined to form the molecules, and from that atoms and molecules, the smooth muscle is formed. That is cellular level. Cells are made up of molecules, and when tissue level, tissues consisting of similar types of cells. Organ levels, organs are made up of different types of tissues. Those are epithelial tissue, smooth tissue, and connective tissue, as just we discussed. And all the organ levels, organ system levels, organ systems consist of different organs that work together closely. When coming to the final one, organism level. The human organism is made up of many organs. Here we will take the human as a model organ to explain the organismal level. structural organization. As just we discussed, the human body is composed of 200 different types of cells with four basic kinds of tissue. Those are epithelial tissue, connective tissue, and muscle tissue, and nervous tissue. A tissue, a group of cells which are similar in structure, which perform common related function. The histology means the study of tissues are generally four basic tissues. One is a systematic, a system by system study, and the tissue is the cells and the extracellular matrix. It is a mesh-like network with proteins, polysaccharides that are secreted by the cells and assembled locally. The organs are formed by the combination of different tissue in variable positions. Here you can see the presence of epithelial, connective, muscle and nervous system. Just now we discussed the cells in the epithelial tissue aggregated polyhedral cells and which is presented small with extra cellular matrix. The main function of epithelial cells are lining of surface or body cavities, granular secretion, connective tissue which supports and protects the body, muscle tissue is for movement, and nervous tissue for the transmission of nerves impulses. As just we discussed, the presence of cells presented in the epithelial is aggregated polyhedral. In the connective tissue, several types of fixed and wandering cells. In the muscle tissue, elongated contractile cells. In the nervous tissue, intertwining elongated process. There are also free cells found in the body fluids. Those were blood and lymph. Organs can be divided into parenchyma and stroma, which is composed of cells responsible for the main function typical of the cells, and stroma, which is a supporting tissue. Except in the brain and spinal cord, the stroma is made up of connective tissue. We also discussed the epithelium cells. Epi means upon, thelial means nipple. So, which is for protection, secretion, absorption, and sensation, that is sensations and contractility. We already seen the epithelial tissue in the last presentation. And what are the common features is a polyhedral form, polarity, which rests on the connective tissue. It contains basal lamina. And here, the things which I want to cover in this PPT is when you see under the transmission electron microscope, the things. So we already covered all the points in the last general histology discussion. So now animal tissue culture. Just up to now, we discussed about the tissue organization, types of tissue, cells are presented, what type of cells are presented in the tissue, and how the movement happens in the uh, organism level, and how cells are formed into tissue. Everything we discussed. Okay. When coming to the our next topic, here I want in this class I want to give some introduction regarding the tissue culture. Tissue culture is the growth of tissues and cells in an artificial medium. Name itself indicating. Here we are. In the last class, we discussed about the biotechnology application. There I have given one example of our Puranas, that Gandhari, who what we call, who damaged her womb, and she broken her embryo into different pieces, and she kept all the pieces in different pots in a sequence of 100 pots, and she also supplied the nutrients to the pots. Surprisingly, after uh, some time, the Kauravas came. So, with this example, tissue culture is there from our Puranas. Okay. Then, when coming to the introduction, tissue culture. Here we are culturing the tissues, a group of cells, but with the help of the artificial medium for uh, producing the entire organ system and organism. So we also discussed about the dolly, the last class. Tissue culture is the growth of cells, tissues in an artificial medium separated organism. Uh, Portable means potent cells. This is typically facilitated via the use of liquid, semi-solid, or solid growth medium, such as broth or agar. When coming to the history of tissue culture, here we have our English physiologist in the year of 19th century. He is Sidney Ringer, developed salt solutions containing the chlorides of sodium, potassium, calcium, magnesium, which is suitable for the maintaining the beating of an isolated animal heart outside the body. In 1885, rocks, rocks removed a portion of medullary plate of the embryonic chicken and maintained it in a warm saline solution for several days, establishing the principle of tissue culture. Ross Granville Harrison, working at Johns Hopkins Medical School and then at the LA University, published the results of the experiments from 1907 to 1910, establishing the methodology of the tissue culture. The cell culture techniques were advanced significantly in 1940s and 1950s to support research in virology, growing viruses in cell cultures, allowed preparation of purified viruses, the manufacturing of vaccines, the injectable polio vaccine developed by the Jonas Salk was one of the first products mass produced using the cell culture technique. This vaccine was made possible by the cell, cell culture research of John Franklin Enders, Thomas Huckel Weller, and Frederick Chapman Robbins, who were awarded a Nobel Prize for their discovery of the method of growing the virus in monkey kidney cell cultures. What are the some frequently used terms in our tissue culture? First one is cell culture. What is cell culture? Cell culture. Cell culture is a process by which cells are grown under controlled conditions, generally outside their natural environment. After the cells of interest have been isolated from living tissue, they can subsequently be maintained under carefully controlled conditions. These conditions vary for each cell type, but generally consisting of a suitable vessel with a substrate or medium that supplies the essential nutrients, amino acids, carbohydrates, vitamins, and minerals, growth factors, hormones, and gases, and regulates the physiochemical environment, pH buffer, osmotic pressure, and temperature. Most cells require a surface or an artificial substrate adherent or monolayer cultures, where other can be grown free floating in culture medium that is called as suspension cultures for aeration. The lifespan of most cells is genetically determined, but some cell culturing cells have been transformed into immortal cells without life, which will reproduce indefinitely without death. Indefinitely if the optimal conditions are provided. What is primary cell culture? This is the first culture, a freshly isolated cell culture or a culture which is directly obtained from the animal or human tissue by enzymatic or mechanical method. These cells are typically slow growing, heterogeneous, and carry all the features of the tissue of their origin. The primary objective of culture is to maintain the growth of cells on an appropriate substrate, which is available in the form of glass or plastic containers under controlled environmental conditions. Since they are directly obtained from original tissue, they have the same karyotype, number and appearance of chromosomes in the nucleus uh, of a eukaryotic cell as the original tissue. Once subcultured, the primary cell cultures can give rise to cell lines which may either die after several subcultures. Such cell lines are also known as finite cell lines, or many continue to grow indefinitely. These called continuous cell lines. Okay. From the primary cell culture, the primary cells are obtained directly from the animal or the human tissue with the help of enzymatic or mechanical methods. So, what is the primary objective for this cell culture is to maintain the growth of the cells on an appropriate substrate available in the form of glass or plastic container under controlled environmental conditions. So here primary cells which are directly obtained from the human, as just I told you, and which gives two types of cells. One is the anchorage dependent or adherent cells, and the other one is anchorage independent or suspension cells. What is anchorage dependent or adherent cells? Adherent cells are those cells which require attachment to the growth, are also called as anchorage dependent cells. In other words, these cells are capable of attaching on the surface of the culture vessel. These type of cells are often derived from the tissues of organs For example, from the kidney, where the cells are immobile and embedded in connective tissue. Encourage independent or suspension cells. Suspension cells do not require attachment or any support for their growth, and also called encourage dependent cells, encourage independent cells. All suspension cells are isolated from the blood system. For example, white blood cell lymphocytes and are suspended in plasma. What is secondary cell culture? Cell culture, primary cell culture, and secondary cell culture. This simply refers to the first passaging of the cells, a switch to a different kind of cell culture system, or the first culture obtained from a primary culture. This is usually carried out when cells in adherent cultures occupy all the available substrate, or when cells in suspension cultures surpass the capacity of the medium to support further growth. The cell proliferation begins to decrease or ceases completely so as to maintain optimal cell density for continued growth and to encourage further proliferation. The primary culture has to be subcultured. This process is called as secondary cell culture. What is the major differences between the primary cell culture and secondary cell culture? Primary cell culture, this is directly obtained from the animals or the plant tissue. And secondary cell culture, which is originated from the primary cell culture. These are the cells which is originated from the primary cell. Primary cells which is closely resembles the parental tissue, because this is directly originated from the animal tissue, animal or plant tissue. Since we are talking about animals, so animal. That's what it is closely resembles the parental tissue. But when coming to the secondary cell culture, it doesn't closely resemble the parental tissue since it is originated from the primary cell culture. And the biological process of the cell may be closer to that in an in vivo environment. Primary cell culture, in primary cell culture, the biological response of the cell may be closer to that in an in vivo environment. But when coming to the secondary cell culture, the biological response of the cells definitely differs from that of in vivo environment. And the other difference is the first cultured cells are derived from the original cells tissue from an in vivo experiment, derived from an existing culture. And the other difference is it cannot be transferred. This can be transformed. This cannot be transformed. Primary cells cannot be transformed, but secondary cell culture is cells are can be transformed. And here, in case of primary cell culture, there is a very less chance of mutations occur. But in secondary cell culture, yes, there is a chance of mutation or genetic alteration of the primary cells taking place. And primary cell culture acquired through steps of raising, dissection, and mechanical or enzymatic disaggregation. And in the case of secondary cell culture, the primary culture is an adherent culture. The first step is to detach the cells from the attachment, the surface of the culture vessel by mechanical or enzymatic means. Then the cells have to be detached from each other to form a single cell suspension. When coming to the primary cell culture, it has the finite lifespan. But in case of secondary cell culture, prolongs the lifespan of the cells. Periodic subculturing may produce immortal cell through transformation or genetic alteration of the primary cell. In the primary cell culture, risk of contamination is very high. It is very difficult to maintain. And the risk of contamination is very low for the culturing of secondary cell culture when comparing with the primary cell culture. It is very easy to maintain. What are other important terms are cell lines. We already discussed about the cell lines and cell strains, but still, cell line, once a primary culture is subcultured or passaged, it represents a cell line. A cell line that experiences indefinite during the frequent a continuous cell line, which will proliferate forever if a suitable fresh medium is provided continuously. Whereas cell lines, cell strains have been adapted to culture, but unlike cell lines, have a finite division potential. A cell strain is obtained either from a primary culture or a cell line. This is done by selection or cloning of those particular cells having a specific properties or characteristics. For example, specific function or karyotype, which must be. What are the concepts in the mammalian cell culture? In the vital concepts of the animal tissue culture. One is the first one is isolation of cells. Isolation of cells are nothing but primary cell production. As just we discussed, by digesting the cells with enzymes, we can produce the primary cells. So cells can be isolated from the solid tissue by digesting the extracellular matrix using the enzymes such as collagenases, trypsinases, and trypsin or pronase before agitating the tissue to release the cells into the suspension. Alternating pieces of tissue can be placed in growth medium, and the cells that grow out are available for culture. This method is known as explant culture, explant preparation or explant culture. And the cells that are cultured directly from the subject as primary cells. And how can we maintain the isolated cells, that is primary cells? Maintaining of cells in culture, for the majority of isolated primary cells, they undergo the process of senescence and stop dividing after a certain number of population levels, while generally retaining their viability, described as the Hayflick limit. Cells are grown and maintained at the appropriate temperature and gas mixture, typically 37 degrees centigrade, with 5% CO2 for mammalian cell. Cell CO2 incubator, and culture conditions vary widely for each cell type, and variation of conditions for a particular cell type can result in different phenotypes. Aside from the temperature and gas mixtures, the most commonly varied factor in cell culture is the cell growth medium. Recipes for growth media can vary in pH, glucose concentration, glucose growth factor, and the presence of nutrients. The growth factors are used for supplementing media are often divided and often derived from the serum of animal blood, such as fetal bovine serum, bovine calf serum, equine serum, and porcine serum. One complication of these blood derived ingredients is the potential for contamination of the culture with the viruses or beyond. So those are the protein materials, yes, particularly in medical biology. And coming to the planting density. Planting density is the number of cells per volume of culture medium. It plays a critical role for some cell types. For example, a lower plating density granulosa cell exhibit estrogen production, while a higher plating density makes them appear as a progesterone producing decalcium cell. Cells can be grown either in suspension or adherent shaken or adherent. Some cells naturally live in suspension without being attached to the surface, such as cells that exist in bloodstream. There are also cell lines that have been modified to be able to survive in suspension culture, so they need to higher than adherent conditions